Thank you so much for such a wonderful introduction. It really covers a lot of stuff already in my talk. And also, I'd love to really appreciate this opportunity, and then thanks for your invitation, your leadership, and the vision to make everything happen. You know, it would be an awesome day and talk about science and collaborations. And I would love to just like share a little bit my journey, kind of like about to demystifying this female bias in our autoimmunity. And also our recent effort trying to develop some novel approach to rewire this self-attacking immune cells, and how can we use that, hopefully in the future, to treat our patients with autoimmunity. So like Dr. Maureen Su's, the slides, we actually happen to have the same slide because we want to highlight how important and how strong the female bias is, right? And then especially in this the autoimmune disease, we have really over like 90% of patients are female, are really suffering. But on the other hand, it might be good for us for females to fighting against COVID-19 or the viral infection. So it's really a trade-off, right? Like, and how do we really demystifying this female bias, how we take advantage of that and to control our autoimmune, but without, losing this important feature to fighting the antiviral infection. So one of the key feature of the differences between female and male, is actually the X chromosome dosage, because that has been all like long basically defined that the patients or like male patients with extra X chromosome, they have like increased chance to develop autoimmune disease like lupus, while the females who have like basically lack one X chromosome have a decreased chance. So it's really suggesting the number of the X chromosome does do something, to our immune system. So how? So back to this, very simple kind of diagram of our two X chromosome in the females versus one X chromosome in the male. Imagine the genes on the X chromosome, their expression level are very different, right? So we have with two X chromosomes, you should have like way more twofold of those gene levels compared to the male. So during evolutionarily, we actually evolved to develop a really important mechanism that's starting from this long non-coding RNA. This RNA in our human is like 17 kb. They didn't encode protein, but they have very important functions to recruit these epigenetic repressors on one of the X chromosome to spreading the whole X chromosome to really shutting down, making sure we have really highly balanced gene expression between male and female. So, and this happens at very early, like embryonic development. We call them X inactivation initiation, which means that every daughter cells of like in our body all have this balanced, this X inactivation. So we want to ensure that we don't have, two difference of the gene expression for our cells in between female and male. But how does this silencing or is maintained through our lifetime, right? Since 1994, the whole field is actually favoring the model, which we call them epigenetic memory, which means that the inactive X chromosome, when they silence, they have this marks of this suppressive demethylation of those KNSM translation, and they can remember throughout the lifetime that this one should be shut down. And however, our recent data really kind of challenging this model, and we basically found that the exist link RNA and their cofactors might be very important to maintain this whole X innovation throughout the lifetime, and to really test whether which model is correct, or maybe they both functioning together, we focus on the immune cell type called B cells. So B cells are, in our body to produce antibodies that can protect from viral infection like COVID-19, but also they can produce some self-attacking antibodies that can, attacking the organs like in your kidney or in the joint that produce in the lupus or disease, or also the arthritis. So we focus on this B cell type, and then we use basically a human female B cell line that with normal karyotype, and we use the drugs that shutting down this epigenetic memory, and also side by side we also perturb this exist long non-coding RNA. So when we apply those in this B cell line, we want to see, okay, which one is important for the maintenance of these X innervation. And surprisingly, we found that actually exist perturbation in the green have a dramatic impact on this X innervation. And you can see a reactivation of on X chromosome, really suggesting that exist long non-coding RNA is essential for the maintenance of the X innervation, at least in B cells. So further looking at the genes, that which genes are silenced by exist, we found a lot of those genes, and turns out a lot of those genes are associated with immune function. And looking further at one of the key genes we call the TLR7, and this gene is very important because it's actually acting as a sensor in our immune system to sense the single-strand RNA that either derived from viruses or can be derived from like dying cells. And basically in the normal kind of cells in our female, we found like the TLR7 can express from the X chromosome and is silenced in the inactive X chromosome. You can see the peaks is only showing up on the active X chromosome. And inhibitors treatment of the epigenetics memory, it doesn't impact these expression. They look pretty similar. However, when you perturb exist link RNA, we see a dramatic kind of like peak here, or showing that expression on the inactive chromosome. And we can see really changing the dosage of this TLR7 if we have this dysregulation of this exist or long non-coding RNA. So this also is very important because it has been shown by a lot of groups that the gene dosage of the TLR7 is highly important and can maybe drive the lupus-like symptoms in the mice. So basically, if you have a higher dosage of the TLR7, the mice will more likely develop the lupus. And in human, and if you have this biallelic expression, again, like just this one from both X chromosome, and they correlate with the increased antibody switching, that's actually good hallmark for basic protecting or us from the viral infection with antibody switching, but it can also be a bad thing in autoimmunity that is attacking our self organs. So it's really suggesting that exist is highly essential to prevent escaping of those X-linked immune genes. And then we're looking further at those genes, and we found this enrichment of the basically two top disease, which is lupus and RA, that's on those genes that escaped from the exist perturbation. And further looking at the patient's data, comparing to the healthy control donors, our lupus patients have this overexpression of the genes that are actually required a silencing by the exist link RNA, but not on the other X-linked genes that doesn't require exist, suggesting that exist is highly essential to maintain this silencing in the healthy control, but somehow in the lupus patients, this is perturbed. So imagine like if we can have this tool to really map this exist dysregulation level at the single cell level and across all the potential disease, then the rationale here is to, if we have this exist dysregulation, we'll imagine it will have this biallelic, like more expression of those exist silence genes on the inactive X chromosome, that's labeled in like the green parts. And then if we're looking at the cell, looking at their gene expression, especially looking at the genes associated in the green, we can calculate like escape score by controlling this independent genes still also on the same X chromosome. And then we reason that the higher, the expression of those genes, suggesting the higher level that exists of dysregulation happens. So we use this strategy. Then we can, using the single cell data that published out there, we first just look at COVID-19 patients, that our female patients actually fighting against viruses are really good at that. And we want to find out like which cells contain this exist potential dysregulation. When we look at a score, we calculate it at a single cell level, like each dot is a single cell. And then the score showing up here, the color, the much brighter or the yellowish, they have a higher score. So we do notice that, again, this is from unbiased searching, we found this a unique subset have a higher expression of those genes, or suggesting there's higher possibility of those exist dysregulation. So what are those cells? It turns out this is CD11C atypical memory B cells. And this escape score enrichment in the cell type only happens in the female patients, highly significant, much higher in this population compared to conventional memory cells, but doesn't show in the male patients. So it's a strong female bias, again, using this kind of human immune data set. But how about our autoimmune patients? So thanks to a lot of consortium that in the effort in trying to profiling our autoimmune patients with autoimmunity, and we find in the lupus on the left, we also see a similar trend. There's higher dysregulation in this CD11C atypical B cells. The same thing in the arthritis female patients. Again, I want to highlight those two are from the patient data and from the B cells that are actually infiltrating in the organ, in the kidney or in the joint. It's really suggesting that dysregulation happens at a very front line, that in this tissue infiltrating potentially or self-attacking organs of the B cells. So then it's really thinking about from basically our single cell data, we are trying to basically generate some hypothesis from there, which leads us to looking at this CD11C positive typical B cells. So what are those cells? Those cells are actually has been found abnormally expanded in female biased autoimmune patients, like lupus and RA patients. And more interestingly is this cell type is highly dependent on TLR7 signaling for their activation and formation, which means, if you remember, we recall our previous slides, TLR7 is on the X chromosome, and it requires the silencing from exist link RNA. So it seems like we can connecting dots, right, from this exist silencing on TLR7 versus TLR7 signaling driving this CD11C atypical B cells. And more interestingly, the female bias is not only just like showing up in the human, but also in the mice. In the aging mice, over 18 months, we see the dramatic, accumulation or expansion of these B cell, unique B cells, only in female, but not in male. And then there's a really beautiful study from Dr. Montserrat's group that's really showing that exist localization are actually completely absent of the focal localization in this cell type compared to the conventional cells. If you see the dot, it's showing the exist localization on the X chromosome. So it's really a lot of this kind of evidences that showing that maybe exist silencing is essential to restrain this CD11C typical B cell formation. So again, this is all kind of speculation correlation. Can we really confirm or validate this hypothesis? To do that, we take the human blood sample from the female healthy donors, and then we did the CRISPR to basically perturb the exist link RNA in those primary B cells. And in vitro, we want to check if those cells are more likely to form these sex-biased autoimmune, the CD11C positive typical B cells. Indeed, we find an increase of these unique B cell type after perturbing the exist. It's really basically validating our hypothesis that the loss of this exist does facilitate the formation of this immune unique B cells. And to really summarize this part, and we can see that, again, from our female and compared to male, we have the evolutionary conserved machinery exist to silencing the one of X chromosome. However, the dysregulation of this exist link RNA may driving abnormal expression or higher dosage of the immune genes such as TLR7, TSL, are all showing up the TLR7 signaling pathway, which potentially drive the formation of the typical B cells. And this is a cell type really correlates with abnormally expanded in the autoimmune patients. So, but to here, we kind of like further trying to understand why this atypical B cells may do something to the autoimmune patients. But before doing that, I want to also highlight this diversity and the specificity of our immune system to recognizing that bad, pathogens or tumor cells, but also there's also a bunch of the B cells or T cells. They are self-reactive, which means they have these receptors like BCR or TCR, specifically recognizing those healthy cells. So with this, basically in our autoimmune patients, somehow these self-reactive B or T cells are dramatically activated, expanded, start attacking the organs. So the current treatment strategy, for example, using the immunosuppressive drugs, is to basically shutting down the whole B cells or even T cells. And we can see that it's not only just depleting the self-attacking B cells, but also they can potentially depleting all the anti-pathogen B cells and also anti-tumor like T cells, which makes our autoimmune patients vulnerable to the viral infections like COVID-19. So this is actually not the thing we want to pursue. And the same thing, for example, if we want to treat the patients with cancer, like immune checkpoint blockade, and those ones will not only just boosting the anti-tumor T cells, can also boosting those self-reactive T cells, also which lead to the immune adverse events, which are very similar to the autoimmune disease. So we are thinking, is it possible if we can only depleting the self-reactive B cells or self-reactive T cells? This is quite challenging, but I think we need a lot of effort to go to this route and to help our patients. So to really focusing on this kind of angle, so I've developed smart viral particles that really take advantage of the viruses. Actually, they can infect the cells based on the receptor recognition. But here, I basically engineered these viral particles as a really programmable or modular platform that we can display any user-defined ligands and also carrying some like protein cargos or RNA cargos and also with the fusogen. So how can we use this to treat or target those specific B cells? What we do is we first can display the antigens. Potentially can be autoantigens or any other antigens on the viral particle surface. And then these antigen can specifically bind to the receptors on those autoreactive, for example, B cells or T cells. At the same time, we also display a fusogen, the engineered fusogen, that upon this receptor recognition, these viral particles can specifically enter into the target cells. So once this enter into the cells, we carry this genetic payload in this viral RNA encapsulated in the viral particle. So when they enter into the target cells, they can drive the gene payload. It can be therapeutic payload, like gene circuits or shRNA, to reprogram the target cells. So here, using this platform, we want to achieve this target delivery of the genetic payload upon highly specific receptor and antigen recognition. And as a proof of concept, we first engineered the viruses, actually displaying this HER2 antigen, and we mixed with on-target and off-target B cells. At the same time, we basically engineer the viral particles carrying a suicide gene. So we imagine the viruses will basically only enter into this on-target B cells, and then those cells will only express a suicide gene, but not an off-target cell, B cells. So when we add a killing drug, we should envision that only the on-target B cells will be killed by the drug, while leaving those good B cells healthy there. And then we do see that a dramatic depletion of those on-target B cells without impacting the other B cells. And the same platform can be applied into the T cells. Basically, we did the same thing, but with the same suicide gene, but we just changed the antigen that can target specific TCR. And we do see a dramatic depletion again here. So this is really showing that again, this is the first step that we want to engineer these viral particles, specifically targeting those potentially autoreactive B cells or T cells to deplete them and without impacting our healthy or good B cells or T cells that can protect us from viral infection. And the last slide is trying to summarize that the most recent effort about the clinical trials or the strategies trying to treat our basic autoimmune patients. Basically, the last few years, there's a lot of clinical trials trying to engineer the CAR T cells, basically like using the T cell, the killer T cells, equip them with this CAR that can recognize like B cell, basically surface marker like CD19. And then these CAR T has been proven very promising in the anti-tumor, in the patients with B cell lymphoma. But here is the same strategy. We want to put those CAR T cells in the patients with a lot of like B cells that can attack our cells. So I think that clinical trials are still ongoing, but there's always more clinical trials, but it seems there's good safety, and also it's a lot promising results, especially if you check their autoantibodies in those patients, they actually completely gone after the treat is the CAR T. But again, these ones will wiping out your B cells, in not only just your self-attacking B cells, but also good B cells. So the next move is to only depleting the bad B cells, or like autoantigen specific B or T cells. Again, like a lot of groups or in industry and also in academia, they're trying to engineer the CAR that can target autoantigen, not only just total B cell marker, but only those autoantigen expressed in those autoreactive B cells, and then to kill them. So I think we'll have looking forward to the more clinical trials or effort into this direction. Again, when we're talking about the accessibility of those treatment to our patients, it's actually quite personalized, which means you need taking the blood or the T cells from the patients out, engineer them, put them back in the patients. It's a lot of effort, time, and the cost. So although it's very promising, but it might be benefit a lot of patients. So the next step is to first identifying those arranging of the pathogenic B cells, and more importantly, can we perform this off the shelf, have the strategy that don't need to taking the cells out, engineer putting them back, instead of directly reprogramming, for example, those CAR T cells in vivo, or specifically reprogramming this or depleting autoreactive B cells in vivo. So that's how the motivation in my lab to trying to further engineer these viral particles. We want to push this into the preclinical models and to see whether the in vivo reprogramming and in an antigen-specific way and as off-the-shelf the feature, hopefully we can have more promising results starting from this. And this is just as my start in my lab, and I want to spend more effort and collaboration with a lot of like bioengineer and also chemist. And we not only just trying to equipping just the antigen specific recognition, but also we want to conjugate the viral particles with small molecules. So can we deliver small molecules into specific cell type to to prevent this whole systemic toxicity from these small molecules? I think that will be a very big step forward. And if we can achieve that, and then we can basically, for a lot of like for a lot of drugs that can fail because of the toxicity targeting so many cell types, if we can equip that only targeting your specific autoreactive B cells or T cells, I think that will be very exciting and hopefully can be beneficial for as more patients as possible. So with that, I'd like to thank my lab members, and most of the work is done by during my postdoc in Howard Chang's lab, and also my collaborators, and also my funding resource. And I'm happy to take any questions. Thank you.